So it is well known that pancreatic beta cell failure drives both type 1 and type 2 diabetes. So the type 1 diabetes basically it occurs because of the autoimmune response and the activation of T cells by antigen presenting cells. The activated T cells produces pro-inflammatory cytokines, nitric oxide, and reactive oxygen species. They actually destroy beta cells, and thereby it results in absolute insulin deficiency and hyperglycemia, which is a hallmark of type 1 diabetes. On the other hand, type 2 diabetes, in case of type 2 diabetes, with the environmental factors like increased free fatty acid and high glucose levels in the circulation tends to destroy beta cells. That ultimately impairs insulin secretion and also causes insulin resistance in insulin responsive peripheral tissues, ultimately results in relative insulin deficiency and also hyperglycemia. So there are several approaches are being done for beta cell regeneration. This is a kind of overview of those regenerative approaches for diabetes treatment. It's like several labs using different molecules and growth factors, including what we have used some of these molecules to improve the proliferation and survival and also function of beta cells, and other approaches like stem cell therapies using varieties of stem cells from different origins to increase the beta cell mass, and also reprogramming both exocrine and also endocrine cell types like alpha cells and SNR cells to reprogram them into beta cells, and also gene editing to correct the mutations in beta cells and also generate beta cells which are resistant to any stress stimuli. So apart from this, stem cell derived extracellular vesicles is a relatively new frontier which is emerging to explore its potential for both regenerative and diabetes therapies. So mesenchymal stem cell is one frontier which is being explored relatively a lot in this area. And this is the glimpse of it. So where you can see that there are studies where they have shown that MSC-EVs induces the immature phenotype of dendritic cells to suppress antigen presentation, thereby inhibiting inflammatory T cell response in islet tissues. And also in another study, what they have shown is hTSC-EVs can enhance, MSC-EVs can enhance the number of islets and improve glycemic control by increasing the regulatory T cell population in streptozotocin-induced type 1 diabetes. And also MSC-EVs carry these varieties of microRNA as cargo, and that facilitates pancreatic functional restoration by improving all these beta cell functional components. And also MSC-EV is known to enhance the function of insulin sensitive tissues like muscle, liver, to improve their function to ameliorate type 2 diabetes. However, MSCs and eventually are known to have many drawbacks like potential heterogeneity, manufacturing challenges, uncertainties in long-term efficacy and safety, and importantly, they attain replicative senescence and also dedifferentiation. So that's where human trophoblast stem cell EVs comes an alternative to explore its potential for regenerative therapies. So far there are some studies where they have shown their regenerative potential and based on the literature. So here just I'm showing some where you can see that hTSC-EVs are able to regenerate and also impart anti-aging effects on skin fibroblasts. And also they could inhibit NF-kappa B and other pro-inflammatory signaling in inflamed epithelial cells. Also, both small and non-coding RNA cargo of hTSC-EVs can induce neurogenesis. In mice, they also can protect sepsis. And notably, this particular study where they have explored that preconditioning of MSCs with trophoblast stem cell derived EVs boosts the proliferation and also regenerative capacity of MSCs. How it does? Those trophoblast stem cells, they secrete EVs with this particular growth factor, which is nerve growth factor, which will be being taken up by MSCs that increases the level of phospho-AKT. Thereby, it causes increase in wound healing, osteogenesis, cell proliferation capacity, and overall the regenerative potential of MSCs. So apart from that, also Accelerated Biosciences in collaboration with NIA Lab have explored the rejuvenating effects of hTSC-EVs and have come up with some compelling data using this particular cell line, which is infrared irradiated, which is a human cell line derived from fetal lung fibroblasts. What they have found is it actually imparts hTSC, both hTSC conditioned media and EVs imparts anti-inflammatory and anti-senescence effects. And also in aged mice, the hTSC conditioned media could suppress key aging and inflammatory markers. So with all these published literature background and also a compelling data being generated by Accelerated Biosciences, we wanted to explore hTSC-EVs can increase pancreatic beta cell regeneration, and which can be used for diabetes treatment. So to explore that, I started with some experiments. I'm going to show some very preliminary data, in beta cells under different models, which are related to both beta cell survival and proliferation. So for beta cell proliferation, I did experiments in both type 1 diabetic and type 2 diabetic models. For type 1 diabetic model, which comprises of pro-inflammatory cytokines, which is a very known model of inducing beta cell stress, and also ER stress using tunicamycin. And I have examined this in human beta cells. Whereas in case of type 2 diabetic model, I have used glucolipotoxicity, which is high palmitate plus high glucose concentration in both aged mouse and human beta cells. To examine its effect on beta cell proliferation, I tested in both human beta cells obtained from both healthy donor and also type 2 diabetic donor. So this is to see whether hTSC-EVs can protect both aged and aged mouse and human beta cells against type 2 diabetic recessive, that is glucolipotoxicity induced cell death. It is very well known that glucolipotoxicity triggers beta cell dysfunction in type 2 diabetes through ER stress, oxidative stress, and inflammation. This is a brief experimental design where I have treated both aged mouse and human islet cells with vehicle and hTSC-EV. Actually, I pre-treated 4 hours before treating with glucolipotoxicity with palmitate and glucose for 24 hours. Then examined the changes in the beta cell death by doing tunnel insulin co-staining. This is the data with aged mouse islet cells. So as you can see here, treatment with glucolipotoxicity, toxic media, it significantly increases aged mouse beta cell death. When we treat hTSC-EVs along with glucolipotoxic media, it significantly decreases the beta cell death, and which is it brings to almost to the basal level. I also looked at the gamma H2X, which is a DNA damage markers and also senescent markers in aged mouse islet cells, as it is the representative picture, where you can see that gamma H2X is in red and co-stained along with the insulin. So this is the data I have so far. You can see that there is kind of increase in gamma H2AX levels upon glucolipotoxic treatment, but hTSC-EV treatment could decrease the increased level of gamma H2AX. It is not significant so far with the number of experiments that I have done. I'm going to continue this. And also I have examined this with human islet cells. This is the characteristics of human islet donors. And this is the data. As you can see here, glucolipotoxicity increases significantly the human beta cell death. That's being significantly decreased by hTSC-EV treatment. So other approach is to do with the cytokine treatment, which is a type 1 diabetes stressor, to examine hTSC-EV's effect on beta cell death. So, and it is well known that pro-inflammatory cytokines triggered beta cell death by inducing ER stress, which is the hallmark of type 1 diabetes. This is the brief experimental design where I have treated human islet cells with either EV buffer or hTSC-EVs, as I mentioned, like 4 hours before treating with this pro-inflammatory cytokines or tunicamycin to induce cell death. Then did tunnel co-insulin staining to measure the level of beta cell death. And these are the human islets that I used, the donor characteristic, the human islet that I used for this experiment. This is a representative picture where you can see that the tunnel and insulin co-staining for cytokine and cytokine plus EV treated condition. This is the data. So just by treating with cytokine, human beta cells are being increased with their cell death, and the cytokine treatment along with hTSC-EV significantly decreases that pro-inflammatory cytokine effect. This also have examined and by treating with tunicamycin, which is a ER stress inducer. So with that also hTSC-EV treatment kind of decreased, but it's not significant so far because of the very small number of experiments. I'm going to continue this also to see whether we can see any conclusive results of the effect. And also I looked at the changes in the gamma H2AX in this case also. Seems like it's being decreased, but as you can see that it is a very small n. So that is where we are right now. Then finally, I also looked at the ability of hTSC-EVs to induce human beta cell proliferation. And it is very well known in the field that the relative rate of beta cell growth or proliferation is high during the infancy, but it gradually declines thereafter to adulthood. And also adult human beta cells are quiescent and are very resistant for proliferative stimuli. So to explore the hTSC-EV's ability to induce these quiescent and resistant beta cells. So I started treating human islet cells with vehicle or hTSC-EVs for 24 hours and looked for the beta cell proliferation level in terms of Ki67 and insulin co-staining. These are the human islet donor characteristics. And this is a representative picture where you can see that vehicle and hTSC-EV treated human islet cell culture co-stained for insulin and Ki67. This is the data that I have so far. This is very clearly, I could see that hTSC-EV treatment significantly increases human beta cell proliferation. Generally, they are not that proliferative in nature. And also, I started looking at the hTSC-EV's effect in islet beta cells obtained from type 2 diabetic donors. Generally, type 2 diabetic donor islets are much more resistant for any proliferative stimuli. I used different concentration of EVs to examine this. You can see that with whatever the concentration that I have treated, Of course, it's just one experiment. Looks like with the higher concentration, I could see more consistent result. And this is the data from two replicate from the same experiment. So I'm going to continue this one to see if there whether we can see any conclusive results in this condition. To summarize my preliminary findings, hTSC-EVs protect both aged mouse and adult human beta cells from glucolipotoxicity, pro-inflammatory cytokines, and also ER stress-induced cell death. And hTSC-EVs able to induce adult human beta cell proliferation, which are refractory to proliferative stimuli. To be done, of course, I need to increase all the N's for the experiments and data that I've shown. And also, this is very essential to do their dosage and time course effect of hTSC-EVs. And also, it's essential to examine the effect of hTSC-EVs on beta cell function at both basal and upon glucose stimulation to examine the beta cell's capacity to induce insulin secretion, to secrete insulin. So with this, whatever the preliminary data that I generated during these tests, we could able to apply two grants. One is on the new investigator or new or early stage investigator grant from HARN, which is about type 1 diabetes pathogenesis and to test potential therapies. And also another grant that is in collaboration with SICO, which is about small business technology transfer. This is, and also which was exclusively for this industrialization and translation of EVs for use in regenerative medicine. So with that, I would like to conclude, by saying many thanks to my PI, Dr. Rupangi Vasavada, for all her support and interest with this project, and also lab members, especially Zelda Cisneros for all her support with my experiments and analysis, and rest of the lab members and people from Accelerated Biosciences, especially Utah for its support, and SICO is instrumental for establishing for this collaboration, and all the other persons who are involved with all the logistics, and Archana, who is very supportive in my grant writing, and our funding agencies, without which I cannot do this work, and also the human islet supply sources, and also finally the human islet donors. Thank you.